And now our next topic and our next presentation. Scientists from all over the world have been trying to develop new drugs, new medications. Biotech is the big buzzword. And during the pandemic, the word of the day was protein. How important are proteins? How are they generated and synthesized? And what does AI have to do with it? Let's hear about it. Head of Bioinformatics Group, research fellow at AIRI, Olga Kardymon. Her presentation is about AI for protein molecule design and generation.
Добрый день, уважаемые участники конференции. Добрый день.
Hello, dear participants and dear viewers. Today I would like to talk to you about a topic that is especially close to my heart. It is very exciting for me. I will try to share my emotions about it and, of course, my knowledge. We will talk about designing and generating de novo proteins, which means proteins that were non-existent in nature previously. We will talk about proteins, what they do, what is their role and function, and where they come from in our bodies. And then we will look at the traditional approaches for protein design. Then we will move on to the role of AI and what does AI do within these existing approaches to protein design. And in conclusion, we will discuss the applications. So what is protein? Protein is the workhorse in our body. Anything that happens in our body is in one way or another related to proteins. Proteins interact with each other, they interact with other chemical tools. The main function of protein can be various. Thanks to actin and myosin, our muscles can contract. Thanks to receptors, we can protect ourselves from virus attacks. Thanks to insulin, we have this signal function in our body, and many more. We have transport function, protection, preservation of different ferments in our bodies, structural function. The list goes on. Let's have a look at this and remember where proteins come from, because you all know it. We have the DNA strand, as you know. It is stored in the nucleus of a cell. Out of the DNA strand, we can generate the RNA strand. With this starting process, we already have protein playing an important role as an intermediary. The RNA molecule is produced from the cell nucleus, and it is connected with the ribosome in our cell, where the synthesis of amino acids happens out of the 3 nucleotides of an RNA. This forms a sequence, a chain, which is the primary structure of any protein. Out of this primary structure of proteins, we get the full-blown protein, which will have the many important functions. What is the difference between the different amino acids? The difference is the residue that have different biophysical and biochemical properties. Some amino acids are positive, some are negative, some are neutral. Some amino acids are able to form hydrophobic globes, some are not able to do it. And if we know the amino acid residue and its biochemical and biophysical properties, from that we can infer the protein and its functional roles. A very important definition that I wanted to show you is the backbone of protein, its backbone structure. On the left, you can see the central structure of protein, the key atoms of hydrogen. Red dots are oxygen, and blue is nitrogen. And then the coding of amino acids. Let's cut out the residue, and then what we have is on the right the backbone of protein structure, which determines the protein that we get as a result. This is exactly the structure that
Now, there can be secondary structure, either the alpha helix or beta sheet. If you talk about LMs, it's like words and sentences. Then the tertiary structure is developed based on secondary structures, and there we have a functional domain, functional protein. Quaternary structure is usually multi-dimensional element that is like hemoglobin or an antibody. So this is quite complicated, a complicated element. Now, what is a computational protein design? Computational means we use compute. If there is a design, it means that we create something, but basically that's about the implementation of the different changes in the sequence of our proteins to get the new folding of the protein and its structure, or to get the requested functions or some biophysical properties that we need. Now, currently, the most popular set of algorithms, the most popular model that is consists of different elements is called Rosetta. It's has been developed in the Institute of Protein Design in Rosetta, and now every designer can select what they need, like the protein docking, or the modeling of the loop that we have in our proteins that plays an important role. So you can download the package that you need, and then you have the magic that is happening. It is quite a complicated magic. It needs some biophysical knowledge and skills, training. Now we get a target in a design. It can be either the entire protein or some functional part, or either the functional motif or some active part. We know something about our our input, and then we start changing the amino acids in the structure of the protein, and we look at the changes in the folding of the protein and how these elements are changed. Now, then we need to make a scoring how different energies, when amino acids are connected to each other, how are they changing in comparison with the input protein that we have analyzed at the start. And our goal is to find the combination that will have the function, but at the same time will be the most efficient. This is when the protein is most stable and saves its functions. Let's look at the pipeline, how these two approaches work together. We're talking about the rational design when using computer-aided design. When we take a protein, finding the mutation that we need, then we take a gene, some mutated gene. We transform that, make the protein expression, and then we purify the protein, and we get the protein we need. The second approach in labs is directed evolution. We not have a compute here. We have random clones of the mutations of our genes, and we start to check them for the stability, for the affinity, selectivity, and compare them with the proteins that we know about. And obviously, the proteins that fit the requirements are being selected. That's great. But if the result is not good, and that it's never good from the first try, then the protein is resent for the production again. But that's a complicated process, therefore. But there are now new ML tools that helps us, without knowing the biophysical and biochemical properties, to get the data sets, and the models can learn the patterns that are needed for the further design of the proteins. And this stage is now became a little bit more efficient, at the same time much more time consuming, and we obviously use much less compute. Now the models that we are using right now, and this is basically the AI finally. Now I will talk about the most popular task in protein design. You need to make some enzymes with more thermostable attributes. If a protein is warmed up, then the structure is changed. But if the amino acids is changed in the protein, the stability of the protein is changed as well. But when we need to take enzymes and select the most active ones, we can try to select the mutations and to find the conformation of the protein that will give us more stability. Now, we have done that, but we have not only selected, we used the language model that has different neuro-free energies. And by training this model, we can say that in the output, with the new mutation, we can predict whether the protein will become more stable or less stable based on these mutations, by analyzing these mutations. Now, you can download the code from GitHub. It's an open source model that is used by in our web interface. And less than a month ago, there was an article published that talks about how we tested the model. This is a very simple tool that appeared right after AlphaFold. AlphaFold won a protein competition, and the first researchers in 2021 tried to use AlphaFund to predict what proteins need what attributes. Now, we take some sequences, the sequences of our amino acids with different mutations. We put it in AlphaFold. Thanks so much to the creators, because there are some metrics for the prognosis of the protein. And when we use the structures, we can then use some evolution algorithm to optimize, or the structure with the right scores can be sent to the Rosetta. In 2021, just recently, that was one of the groundbreaking approaches. And this is about automation and improving the attributes. This is a very challenging process in terms of the resources, because the first AlphaFund has an algorithm that takes quite a lot of time. It's a very complicated procedure. Now, the Institute for Protein Design made their own model. It's called RF Design. And in this model, they showed that the design can hallucinate on the protein. But hallucination in protein design is actually good. Why? The reason is simple. We try to create a structure that our model didn't know about before. We are making a new protein that has never been known to nature. But it's very important for these hallucinations to have biophysical, biochemical properties. So the hallucination in generation of new proteins is good. So to hallucinate, the RF design in their approach using the algorithms for the new mutations in the random sequence that it is learning. And we have a 3D structure as the output that has some conformation and that is similar to a natural protein.
Но как же нам сделать так, чтобы нам модель генерировала белок, нужный под нашу задачу?
This is how we can generate the protein based on our task, and our colleagues published an article in Nature, and this means that we can read the text of the proteins as the text of any other sources. If we can make some tags like about politics or sports, we can put the same tags for the protein sequence and the protein text. And it will show the family of our proteins, and the family of the proteins will predict the function of the protein that is going to be generated. Now, 180 million sequences was used, and 19,000 families were added that are described in PFAM database. It was fine-tuned, and the results were very exciting. They were able to generate 1 million sequences that look like the lecithin protein. This is the antibacterial protein that can that is present in the saliva and other fluids that we have, and to have a lecithin from 1 million of sequences, there was hundreds of sequences that were checked, and are using biochemical methods. Only 72 proteins were well expressed. So this is on paper. It works on paper, but can it be translated to the real world? Well, that's a question. A significant part of that proteins showed some activity. So they were related to they were showing that they actually have some antibacterial function. Now, it was decided to synthesize them, and this is what they do on the pharmaceutical companies. And from 5 synthetically synthesized proteins, only 2 were well expressed in E. coli cell, and they both have catalytic activity, so they were destroying the bacterial walls when they were checked in terms of their functionality. And one of these 2 proteins was checked with the structural analysis, and yes, it was said that it is in line with the predicted structure, and it is similar to the structure of protein that is lecithin protein. Now, in biology, there is another task that is very important, the reverse task, not only to predict the sequence, but when we have some framework and we understand that we need it to work with a protein, and then we need to put this framework to the sequence to synthesize it to the protein. And last year, there was a model which is called protein MPNN. It is based on a very famous model for working with text, message passing network, and it is reading the proteins. There are 3 layers of encoders, 3 layers of decoders, and our model gets some coordinates based on some framework, and these are where we have all the nitrogen and nodes and other things, so that we understand what should be the protein that we should generate. And this model helps us to use some sequences to get the amino acids that are important for us. And this is what we use to work with our protein, synthesized protein. And based on that, the sequence that is forming our protein will be generated. A very good indicator for our model is as follows. Is this one, protein MPNN, but it recovers the native protein backbone with a 52% efficiency, unlike 33% efficiency for Rosetta. And the time is much small. The time frame required is much smaller, and actually, in terms of the stability, these synthesized proteins are more stable comparing with native metrics, native proteins. Now, there are many things under the hood of the model, but anyway, the results are quite promising. The diffusions of the proteins. Now, the diffusion models is the state-of-the-art models, and if there is no diffusion, I mean, we would put the data sets and we would say diffuse it, to put the task of diffusing it and to show what is there. It works with the images, but proteins are different.
In order to create Gaussian noise to generate the proteins, we need to first handle the coordinates. We take the coordinates, the range between the atoms, we add the Gaussian noise, and then thanks to the Brownian motion directions, we can generate it back into the protein structure. This summer, a model was created in Protein Design Institute. This model is based on its own Rosetta fold model, which takes the sequence of amino acids as and also some homologous templates. And then it takes the initial coordinates to predict the protein structure. To create the AIRI diffusion, they switched the approach somewhat. They masked the input layer. They added self-conditioning, the step between generations which repeats itself, and then the diffused coordinates that result in the protein structure. What is impressive about it is that the colleagues were training it based on the protein database, PDB, which includes 3D structures of proteins, and they also used Rosetta fold proteins. And they produce good results with the conditional protein generation. So we get the clear alpha spirals in proteins, but they can add other conditions, and so they can generate symmetrical proteins that can use to produce vaccines, or they can also create different binders, the proteins that bind with other proteins. They can also create protein scaffolding. So the capabilities are limitless. Let's have a look at how they visually represent their models. On the left-hand side, we see the diffusion of a symmetrical model, which can be used as a nanoparticle for nanogens, for example. And on the right-hand side is the insulin receptor protein, which can then be used for different metabolic problems. The RF diffusion generates sequences that are 600 amino acids long. The colleagues say, frankly, in their paper that they can probably generate more, but 600 amino acidic residues, that is the length that they can verify using AlphaFold or Rosetta Fold or other tools. If we calculate the proteins, 20 amino acids in our bodies, so 20 amino acids to the power of L. And what we have is a 4.37 to the power of 781. That is the amount of proteins that can be generated. That with the 212,000 in the protein data bank, that is very few. As for the UniProt sequence database, there are 251 million of them. So in this universe, we are only playing in a very small corner of it. What can help us create new proteins unknown in nature, moving from 3D structures to sequences? If the RF diffusion takes the 3D structures and coordinates it knows, the new model developed by bioinformatics group at AIRI by my colleagues, together with the Higher School of Economics. This model is called Dima, after our legendary colleague. So this model is based on creating noise out of ESM2 embeddings, the language model for proteins, and then out of that noise it generates new proteins. What are the advantages of Dima? Dima has several advantages. The first one is that it is based on the ESM2 embeddings, which means it knows more encoded context than there is in the coordinates, because, for example, there are parts, IDRs, which are not very well structured in proteins. These IDRs are almost always lost when a crystal is built. to determine the 3D structure of a protein. But if they are there, we know that they can that the model can generate them. Our model is faster because it does not have the 3D coordinates, so for the same reason. And interestingly, for now, we only have this model for non-conditional generation, and based on the based on its generations, we can see that it generates functionally significant proteins related to different families. So we can compare them with what other models can produce, and we compare them with a data set. So clearly, what we are generating is something that is practical. We have not done much in that project yet. I hope we will get back to that after the New Year holidays, and I will tell you more about it. But right now, I wanted to discuss with you the practical applications, why it is all needed. Why do we need this? Now, especially after the COVID pandemic, you would think I would be talking about drugs, drug design, medications. Yes, of course, but protein is used in many other industries. For example, there are enzymes which need to be improved for them to be able to recycle waste, which have catalytic activity for waste treatment, waste recycling. There is another biotech cluster which creates substances, for example, to improve the properties of a detergent. But anyway, all these applications, in addition to drug design and antibodies, agricultural needs, household needs, or ESG, they can be divided into 4 big blocks. The first block is where we generate the scaffolding for the protein, the active site of the protein, to ensure proper functionality of that active part. Second, we know the backbone, but we do not know what it consists of, so we need to synthesize it. We synthesize the amino-acidic composition in order to be able to then synthesize the protein in bacteria and for it to have catalytically significant functions. Third, designing protein fragments. The antibodies on their surface have this non-structured loop, which is very difficult to recreate with the existing tools. But this is exactly the loop that interacts with the virus surface and important sites. We can generate the loop separately, leaving the backbone only as a functional element, thanks to modern generative models. And of course, the fourth application, a huge wealth of opportunities there, the de novo generation. I'd like to quote my colleague here. He created a protein design group in the United States, and out of that group, a whole institute emerged. Same as all the other protein designers, he thinks that artificial intelligence and AI-based tools are capable of transforming medicine. We will be able to create new compounds, new drugs, which will be more narrowly specialized, more targeted, and which will have the properties that we need. At this point, I would like to thank you for your attention, and I also wanted to thank my colleagues who are part of my team working on protein design, and I also wanted to thank Dmitry Vetrov and Vyacheslav Mishininov from the Higher School of Economics, who got carried away with this idea of protein design and then created something wonderful with it. Thank you very much. And we thank you, Olga, for this interesting and informative presentation. As usual, a couple of questions. If you have the time, of course. An interesting question here. I was listening to your presentation very avidly. I understood hardly anything, but that was very exciting. Clearly, I have some gaps in my knowledge about AI, and I will be rewatching the presentations. You can do the same at aij.ru. So the question is, what are the limitations in using AI in protein design? And what are the challenges? Well, the challenges are creating new molecules which will be safe and tailored to our needs. And the restrictions today is verifying these models, checking them. Each compound that we generate needs to be validated. We need to verify its structure, its safety, its efficiency. So we are just at the beginning of this path, at the beginning of this journey. And as measurement, you take detergent. Okay, I like that you remembered that example with the detergent. No, of course not. But we do have lots of tests for our models with different materials, analyzing their synthesized properties. Thank you very much, Olga.